Welcome back. I'm Dr. Leslie Blankenship-Williams, and in this lecture we are going to finish our discussion on the history of microbiology. So where I left off was talking about the theory of spontaneous generation, which we know to be false. And where I left you with was this idea that Francisco Redi's experiment in the 1600s definitely disproved the theory of spontaneous generation for macroscopic organisms like flies, but people still held on to that belief for microscopic organisms, simple stuff like bacteria. And so, again, we know that that's not true. We know that the theory of spontaneous generation is not true, but it took another 100 and something years before it was formally and finally disproven once and for all. And the man who did that is named Louis Pasteur. So you may recognize his name. He is famous for inventing the Pasteur pipette and also pasteurization, the idea that your milk and orange juice and other liquids are heated to about 60 degrees to kill most microbes before putting them in the refrigerator for consumption. Louis Pasteur's most famous contribution to microbiology is actually disproving the theory of spontaneous generation. So it is important to understand that we need to get rid of that theory before we can embrace the true germ theory of disease and the idea that bacteria come from other bacteria, rather than just air, water, and nutrients in some magical combination. Because if we understand that bacteria come from other bacteria, and I have a bacterial disease, then you understand that you can only get that bacterial disease from bacteria that are maybe coming from me, rather than some magical combination of air, water, and nutrients that you're exposed to. So, disproving the theory of spontaneous generation was absolutely instrumental to embracing the germ theory of disease. So let's take a look at Louis Pasteur's ingenious design that allowed him to formally and finally and convincingly disprove spontaneous generation. So Louis Pasteur was an expert glassblower. He did a lot of stuff with glass, hence the Pasteur pipettes. And what he noticed was that broths and other like orange juice or apple juice or meat broths or stews would become contaminated after being heated up just from exposure to the air. And people said, well, of course that's what happened, because the broth was exposed to air. It's got water in it. It's got nutrients in it. So bacteria spontaneously generate. He wasn't so sure. So what he did is he would take a broth of some sort and he would extend the flask neck into what is now known as a swan neck. And then he would heat the broth. And of course, heating the broth would kill everything in it, and so the broth would remain sterile. And what made his design so ingenious, the swan or S-shaped flask, was that air could still get in, but yet it didn't become contaminated or spoiled by bacteria. So it still had access to air, water, nutrients, but wasn't getting spoiled. And that is because we now know that such spoilage happens when dust particles containing microbes fall from the air into the pot. And here, the dust particles were getting trapped in the U-bend. So another way that he could show that it really was dust particles in the U-bend is he would take some of his sterilized broths, show that no contamination happened, no spoilage happened, and then he would take the flask and start doing this, and the air would start wishing the bacteria that had settled in the U-bend into the flask and then spoil it. Another thing he would do is he would just take one of his precious sterile flasks with broth that was unspoiled and just basically knock off the top of the flask, and now you have a broken neck on that flask that is directly exposed to the air particles falling down. They would become spoiled, etc. Here's another picture of one of his famous swan neck flasks. So that experiment decidedly refuted the theory of spontaneous generation once and for all. It died and has remained on the pile of ashes ever since. But that still didn't prove that germs cause disease. Now, Louis Pasteur was definitely dabbling in that area, but he wasn't able to prove it. He was only able to show correlation. And as you know, correlation and causation are not always the same things. So how did he correlate diseases with particular bacteria? Well, Louis Pasteur was actually recruited to investigate the French silkworm industry. And the silkworms, which are worms, would basically get these diseases that would ravage entire factories worth of silkworms, and they would die. And what he noticed was that certain silkworms that showed certain diseases always showed the presence of a particular microbe, like a fungus or a protist. And so he certainly suspected that the protist or the fungus was causing that disease, but he couldn't prove it. So because he couldn't prove it, he was not technically credited with being the father of the germ theory of disease, but his involvement was critical, in that he certainly noticed a correlation and believed that these silkworms were dying from these infectious microbes that were being transmitted, and then, of course, disproving the theory of spontaneous generation, which was instrumental to getting rid of that so that we understand that bacteria come from bacteria. So who did prove the germ theory of disease? Well, it turned out it was Louis Pasteur's arch nemesis, Robert Koch, who was a German scientist. French and Germans, different cultures, big culture clash there. Robert Koch proved it. And Robert Koch proved it because he was able to invent particular culturing techniques that are still used today. So let's investigate what Robert Koch might have been up against. Imagine for a moment that you have a patient with strep throat. So most of you have probably experienced this particular disease. We now know that it is caused by a bacterium known as Streptococcus pyogenes. And when somebody has strep throat, their tonsils are incredibly inflamed and they tend to have white spots in the back of their tonsils, as you can see here. So how would you prove that this person or any person's strep throat was caused by this bacterial species? Well, what we would do today is we would take a swab, and everybody that presented with these symptoms we would swab. And then what we would do is we would take that swab, which should have the bacteria that cause that disease, and we would basically transfer that swab onto a petri dish filled with agar. And this is a blood agar plate. You'll be exposed to it in the lab. Now, what that's going to do is that's going to go ahead and transfer that bacteria onto that plate. Well, anyone could have cultured bacteria, but one of the problems is your mouth has like 300 different species of bacteria in it. So how do you know that you're culturing the causative agent of strep throat instead of something else? And the answer, up until Robert Koch, was that you couldn't. In other words, they didn't really have any way of creating what is called a pure isolate, one species. Because when you would do this technique of just kind of transferring it over, bunches of stuff would grow. And so you'd get all kinds of different things in there, and they would all grow together. So Robert Koch developed what was actually two different methods, but the method for creating a pure culture, one of which is called the streak plate methodology, which you will be learning about and practicing in lab. So streak plates are a way to isolate individual colonies from a mixed sample, like the mouth. Okay, so let's say that you could do that. Let's say that you could create a pure culture. Then what? Then you would create that pure culture and you would want to throw it onto the tonsils of somebody who is not infected with strep throat. And then that person should get strep throat. And then you should be able to re-isolate the bacterium out of that newly infected person. So these set of steps, finding the bacteria in all people who show the symptoms of that disease, isolating that bacteria, creating a pure culture of that bacteria, inoculating that bacteria into a new person that does not have the disease but could get the disease, seeing the disease show up in that new person, and then re-isolating the disease from that newly infected person are collectively known as Koch's postulates. So Koch's postulates are listed here, and you can go ahead and take a look at them. And then the streak plate technique, where you can see the bacteria run into each other on the bottom, but as you go around the top of the agar, you can see that the dots get farther and farther apart. Each individual dot is known as a colony, and one colony represents one cell. So if you can get an individual colony all by itself, that is the basis for forming what is called a pure culture. So this is a necessary step in creating and isolating individual species of bacteria from a mixture like from the mouth. So once Koch's postulates were established in the 1800s, this basically opened up the golden age of microbiology. From here, people were in a mad rush to basically figure out the causative agents of all different types of species. Robert Koch himself determined the causative agent of anthrax, tuberculosis, and cholera, all caused by bacteria. But what about viruses? Viruses cannot be seen with a microscope, and they can't be cultured by traditional techniques like growing them on agar. Viruses were discovered in the very, very, very late 1800s, and through some specialized techniques, versions of these postulates were also applied to determine the causative agents of viral diseases as well. So over the next 50 years, most agents for the infectious diseases of that time were identified. Ones that were emerging or really hadn't shown up yet, like Ebola and AIDS, those were discovered later as they became prevalent in humans. But at the time, they pretty much had it covered. So what does that do? Well, that basically gives us some tools to implement preventative human public health measurements. Like, for instance, if we know that tuberculosis is spread by bacteria from the lungs, and so therefore anybody who's spitting is going to be transmitting, that tells us that we should not be having cans where people could spit. They're called spittoons, and it used to be a thing. People would chew tobacco and spit in a spittoon. Getting rid of spittoons ended up reducing the transmission rate of tuberculosis. But of course, the other component of dealing with infectious diseases is curing them. So bacterial infections can be cured by antimicrobial drugs that are more commonly known as antibiotics. Viruses, the word cure for virus is a little bit more squishy and not as easily applied. Viruses can be treated often by antiviral drugs. Wouldn't say that they cure you the same way that antibiotics cure bacteria, but antivirals can be used to help treat and at least help your immune system out in dealing with the viral infection. Coming into the 1940s, most of the really, really deadly infectious diseases that were known were caused by bacteria, not viruses. The 1918 Spanish flu notwithstanding, so that's kind of an exception. But when you think about diseases that ravaged human civilizations, you have syphilis and plague and tuberculosis and pneumonia and infections after you get wounds or, after from war. Lots of people that were participated in World War I died of their wounds, not because of their actual wound, but because their wounds would become infected with bacteria. So people really were fearful of bacteria. So the first person to discover the antibiotic made major strides in combating bacterial infections. And this actually was a happy accident of the Bob Ross variety, but much more meaningful. It wasn't like a happy painting accident, it was a happy accident. A physician by the name of Alexander Fleming was researching strains of Streptococcus, and he noticed a contaminant on one of his plates. Now, contaminants are very common. You open a petri dish up and a little fungus from the air lands and forms basically what looks like mold on your dish. And he had a plate that was supposed to be pure streptococcus, and it had a mold contaminant, and he was about to throw it in when he noticed something really weird. He noticed that where the fungus landed, there was a zone around it where the bacteria would not come into. So you can see that in this picture here. The bacteria are all on the outskirts of the plate, but they won't grow close to the fungus. And he was about to throw it away, and then he went, that's weird. There's this zone of clearing where the bacteria won't grow around the fungus. What's going on? So he postulated that the fungus must be making some sort of chemical that's leaking into the agar that kills the bacteria or otherwise prevents them from basically encroaching on the fungus's space. And he sent it to chemists. Chemists isolated the chemical and named it penicillin. They learned how to synthesize penicillin, and then the first antibiotic was mass-produced in the very late 1930s and publicly available in the 1940s. And with the advent of antibiotics, we felt like we had a good handle on bacterial infections globally. And I would say that the pendulum swung more towards the deadliness of viruses once we got a really good handle on bacteria. Now, of course, that handle is still a little bit of a slippery slope because we have antibiotic resistance as being a real problem. If you already work in a hospital setting, even pre-COVID, you might be familiar with MRSA or C. diff, Clostridium difficile, as being particularly antibiotic resistant. In fact, there is now a strain of gonorrhea that is incurable. Fun. What we used to think of as trivial, just throw an antibiotic at a bacterial infection and you're done with, now is not so much a sure thing. Combined with the emergence of viruses and our inability to create cures for viruses, we still have a lot of problems with infectious diseases. And I'm sure anyone who's lived in the last year knows this to be the truth. All right, that concludes our foray into the history of microbiology. Thanks for listening.